honor because we are doing everything has science behind. And in my opinion, this is a dream. And you are, yeah, the guy of this dream. Please, your time, take your time as long as you want. Big pleasure to have you here.
Well, thank you so much for hosting, and I really enjoyed the opportunity to speak to you all. I must apologize. I really have to speak Portuguese, so all this is gonna have to be in English. Maybe I can work on this as we develop.
Yeah, sure. I'm sure you're going to speak much better than me.
So what I'm going to be doing is in my course and here today, I'm going to be making a case for an evolutionary perspective on understanding biodiversity and deploying biodiversity to solve some of the big world problems and promote equity worldwide. So one thing that I would like to make a pitch for, if you'll indulge me, is for some of the cool research and training that's going on at the University of Georgia in the plant sciences. We've got this plant center. It's an umbrella organization for people doing research in our College of Agriculture, our College of Arts and Sciences, forestry, ecology, even engineering. Okay. And it's got about 60 labs, 60 different PIs that have their labs participating in this. We've got a couple events each year, a big retreat that's an overnight event, which has got one of the most exciting poster session. We had good talks, but student poster session. It's a lot of fun. Let's see if I can figure out how to do this. Let's see, this is good. There we go, and then the.
We have a very analogical pointer here.
Excellent. I like that. I like that. Yeah. And so, yeah, if you're interested in coming up to the US, definitely you're always welcome to come and visit us in Athens and check out the plant research going on at the University of Georgia. There's quite a bit, again, everything from very applied work in the College of Agriculture, biotechnology, to more esoteric work that I was trained to do as a student. The other thing is we've got this integrated plant science program for graduate students. So if anyone is thinking about a PhD program, I encourage you to go check this out. Students will be able to come into the program. They don't have to choose a PhD advisor or even a department immediately. They can choose among all of these departments, all these PIs across doing various types of plant science, rotate the first semester and then jump into one of the programs into one of the labs after having some experience through the rotation. So it's really I think that we get a great bunch of students coming through there, and I think we provide a great experience for incoming students. All right, so that's my pitch on the University of Georgia. Now we jump on into again this argument I'm going to make for the utility of evolutionary thinking and phylogenetic thinking in any type of life science, any type of biology. So I think that many people might be familiar with this first quote, Jeff Chansky back in the 70s, nothing in biology makes sense except for in the light of evolution. In a lot of folks independently, we're not sure who was the first one that coined this term. A lot of it came to mind that nothing in evolution makes sense except for in the sense of in the light of phylogeny. I understand that the population geneticists in parallel came up with the same quote, nothing in evolution makes sense except for in the light of population genetics, and probably a little bit of both, as I've been talking about in the class. This is a continuum, phylogenetics to population genetics. Anyway, this tree thinking was, really integral to Charles Darwin's thinking as he's going around the world and, at the age of 20 something, and encountering all this beautiful biodiversity, and sketching his notebook and thinking, oh, well, what is, why is it that, these guys look similar to each other, these guys look similar to each other? Maybe shared ancestry can explain some of the patterns of diversity that he was observing in his trips around the world on the Beagle. Jump ahead, about 150 to 200 years later, and we're in the age of genomics. And so, but we're doing basically the same thing. We're using genome scale data to resolve relationships, to draw these trees, and then using these trees to think about what, this is a historical context for thinking about how the genomes and the gene families within those genomes are evolving. And so this is kind of my bailiwick. So I'm going to start by very briefly kind of giving you an example of the way in which we've been using genome scale data to resolve relationships among all green plants. And this is this 1,000 plant transcriptomes project. We started this project in 2009, 2010. Really kind of didn't get going until 2011, 2012. But Gang Kashu Wong was at the University of Alberta. He also had a relationship with folks at BGI. He was an advisor for Beijing Genome Institute. And we'll get you're going. Okay. And so he thought, well, I want to do something really audacious. Kind of the vertebrate genome folks were thinking about sequencing a bunch of genomes with plants that might be a little bit intractable because, as some of you all know, plant genomes can be really massive in size, and pine trees, have genomes that are up 10 times or more the size of the human genome. And so let's start with transcriptomes and lots of species from across the green tree of life. And he reached out to me and lots of other folks that could help him with sampling and analysis of this type of data, and culminated in a paper that came out in 2019. This is the workflow that we had, is that, get our material, again, everything from algae to angiosperms. We chose vegetative tissue, that's a shared tissue type in all of these organisms. Extract their RNA, sent the RNA off to BGI in China, Shenzhen, China. They sequenced it, made libraries, sequence, generate data sets for all 1,000 plus species. They actually had to modify their genome assembler to deal with transcriptome data. So for this project, they made soap de novo trans to assemble the transcriptome read data into transcript sequences. Then we clustered those in the gene families using OrthoFinder. So Natalia can tell you all about OrthoFinder. And then once we had our gene family circumscribed, we were able to draw on some computer scientists that were working on ways of doing massive analysis, working on massive data sets for making multiple sequence alignments and gene trees for individual genes, gene families, and then taking all of those trees made for the gene families and making the inference about species relationships. So one thing is that this project really increased the diversity of sequence data in the public databases. So this is NCBI before 1KP and after 1KP, and you can see that there are some groups where we really massively increased the availability of data. Ferns, for instance, nothing there before 1KP. to a lesser extent, but significantly to the bryophytes, and so on. So this is one of the things that this project did, is really contribute to our understanding of diversity, protein biodiversity. And the other thing is that we took these genes and used them to generate a species tree that included over 1,000 species, like 1,100 some odd species, 400 different gene families that seem to be single copy in most of the things that we had. Analyzing those single copy genes was less complicated than trying to analyze big complex gene families. We took all those gene trees and made a tree for the all green plants, again, from algae to angiosperms being the most diverse group. That's cool. The other thing that was really cool to me, in addition to the fact, we resolved a few controversial parts of the tree within the gymnosperms, figuring out the relationships of various gymnosperm lineages. We kind of resolved some issues, figuring out the relationships of bryophytes. We resolved some issues, but some things were really interesting. We'd identified parts of the tree where, even with all these data, lots of samples, lots of gene sequences, the tree was not bifurcated. End up, we have a polytone there in part of the tree where we're not able to resolve species relationships in a bifurcating tree, in a tree-like fashion. And to me, that's really, some people were disappointed. Oh, well, go back, work over, do another analysis. I'm like, no. This is telling us that there are times in the evolutionary history of plants where we have very rapid diversification. It turns out that this point, it's the last common ancestor of core eudicots. That ancestor was a hexaploid, and so that hexaploid spewed off a bunch of lineages very rapidly, and again, now we try to use these data to resolve the bifurcated tree. We just can't. Getting back to the diversity, these data were also used by biotech folks, by bioengineers, particularly some of the genes from the green algae. Mined those data, found that there were these channel rhodopsin proteins that were already from, one or two of those from green algae were being used for optogenetics. That is, using light to turn genes on and off. But before 1KP and discovering the diversity of channel proteins that exist in algae in particular, really there was only one wavelength that was these channel rhodopsins were not really able to tune in to multiple wavelengths to make it so you could have an on and an off switch, for instance. And so in this the channel rhodopsin proteins that they found in the 1KP data set, and these were from algae that Michael Laconian was the curator of the algal data set in collection in Germany. He provided these algae. Ed Bowden at MIT mined the data. We generated the data, Ed mined the data, and developed assays to figure out, well, what are their absorption characteristics, and it was able to find these different channel rhodopsins that had divergent absorption patterns, and so was able to now have a two-channel system that would allow them to turn things on and off with one channel rhodopsin or the other. So again, biodiversity, protein biodiversity in the world is of use. This technology is now used very broadly for human biology and plant biology too. I'll come back to that point very long. So another big project that I've been involved with and kind of helped lead is this Open Green Genomes project. So we start off with the transcriptomes. We go back and look at the genomes. Now, of course, the transcriptomes it's a subset of the protein coding sequences that are in the genome. Doesn't tell us anything about the structure of the of these genomes or genome evolution, really, having transcriptomes. So let's go and look at genomes. Went and looked at the availability of genomes at the time, and we saw that there were big parts of the land plant tree of life represented with these purple tips here, these purple taxa. So for instance, over here, ferns, at this time when we started looking at this, there was only one fern genome available, but there's this huge diversity of genomes of species within ferns, and no genomes available at the time we started this project outside of the water ferns and zoa. Really the same thing for the gymnosperms, monocots, non-grass monocots. They're poorly represented in the availability of genomes at this time, and so we thought, okay, let's have a project, and JGI just signed on this idea. Let's have a project where we're going to go and sequence genomes from across the land plant tree in parts of the tree where there aren't available high quality genomes. Now this is a big project again, just like the 1KP project. It's kind of organizing together the large community of folks. We're always open to having more, so I actually be happy to talk with you about what are we missing, how might you be interested in helping us analyze the data that we're generating. But the folks that are really doing the heavy lifting are at the Joint Genomes Institute in California, Hudson Alpha in Alabama, Arizona Genome Institute, and these are folks that are we provide the material, they extract high molecular DNA, make libraries, do sequencing, put together the assemblies and annotations, and then give it back to us, and we got to figure out the biology. If you want to go and learn about where we are with this, so right now we've been lucky enough to JGI's allowing us to have some mission creep. So we started with 35 species on our list. We've been able to add a few species as we've been showing progress using the data that they're generating. So anyway, if you want to go to find a zone, you can go to our project, Open Green Genomes, and find genome sequences that we've generated thus far.
But no agave.
Agave was generated also by JGI. It's available on Phytozone, but that was a project being led by folks at Oak Ridge. We're involved with this project, but not as part of the genomes. I'll talk about that. Hold on. Here's our first publication, came out in 2022, a fern genome. This is sea fern, so it's a very widely used fern for teaching purposes and experimental purposes. It was the first genome for these homomorphic ferns, and so we did various types of analyses. We found that it is a really dynamic genome. So there's a genome duplication event that occurred early in the history of the genus, and found that genome duplication event that the two subgenomes got scrambled wildly. So there's no synteny between the two subgenomes at all. But some other thing we found is that there's multiple instances of horizontal gene transfer from bacteria and fungi, actually. And some of these genes that have come in these pads, for instance, are really important for contributing pathogen resistance for serotolerance. Another project that we're pretty far along on, the manuscript getting ready to submit it right now, is analysis of some of these members of the Poales. So again, grasses have been well sequenced. Things just outside of the grasses, Joinvillea, for instance, Ecdyocloa, flagella, no sequence available for those yet. So we've added Joinvillea and collaborators added Ecdyocloa, and we now are analyzing, well, what happened following this genome, this whole genome duplication event in the last common ancestor of all of the Poaceae.
Sugarcane is also not there.
Sugarcane is also not here. It's a very, yeah. I guess you guys are working on it. That's a very complicated one with all this.
No, it's a good news because we are going to include, yeah.
Good. With these genomes, what we're able to do, is Hiroshi Maeda was working on this lignin biosynthesis pathway, and in grasses, they are able to make important part of lignin biosynthesis is making this chimeric acid, and most plants are doing this with p-tal that's converting tyrosine into a PAL, I'm sorry, converting a tyrosine into cinnamonic acid and into chimeric acid. What the grasses are able to do is they're able to use this this p-tal to convert tyrosine into chimeric acid. So they're able to do both of these things. So when did that arise? Their first guess was, well, what this may, these two duplicates may have arisen when in the last common ancestor of grasses with this whole genome duplication. But our genome data very, very clearly shows that no, that's not the case. Pineapple only has one copy, the PAL, an ancestral copy of this genome. In Joinvillea, not in the last common ancestor of Joinvillea and all the grasses, there was a tandem duplication event. And then, following in the whole genome duplication event that occurred later, both segments of both chromosomes are maintaining these two duplicates that track back to this initial tandem duplication event of PAL and p-tal. Immediately following this, apparently from analysis that Hiroshi's lab has been doing, following this genome duplication event, last common ancestor Joinvillea, Ecdyocloa, and the grasses, it seems that the p-tal's function, we're looking here at p-tal, it is able to work both with again tyrosine, so it's got tal activity, and phenylalanine, so it's got PAL activity. So and this is an ancestral function, but shortly after this duplication event, the p-tal copy is able to process tyrosine. So anyway, these are examples of ways in which we can use genome data, maybe add genome data to the data that's already in available databases like Phytosome, other databases available, genome databases that are available. Organize our gene families, make gene trees, maybe refine those gene trees with some SNP information, and provide gene trees where we're annotating those gene trees with timing of gene duplication events, and then asking, well, following duplication event, how do these duplicates, are they retaining the same ancestral function? Is there evidence of sub-functionalization or neo-functionalization, as we saw in the p-tal example? And then, use that information to design new experiments, to do, design new organisms, to do synthetic biology. So really ways in which we want to zero in on gene function, put together genome data with understanding genomic variation and phenotypic variation, make that genotype to phenotype connection is by doing comparative genomics, including either genotypes within a species or very closely related species. And this gives us an opportunity to look at if there's differences in phenotype, what are the associated differences in the genomes? So I give a couple of vignettes where we're doing this sort of thing. The first one is looking at where the objective is to understand something about the evolution of CAM metabolism, or cressularia acid metabolism in plants, generally using yuccas and agaves as a study system. So you probably all know, everyone knows what CAM metabolism or what CAM is. Plants are opening up their stomata at night, pulling in carbon dioxide, then fixing that carbon as carbon acid, and then doing photosynthesis when the sun is out, but when the stomata are closed during the day. So, here are some of the key proteins that are involved in this process and are active either at night. PEP carboxylase is the protein that's doing the primary fixation of carbon dioxide, making malic acid, getting that into, then that has to get into the vacuole. Then during the day, that's got to come out of the vacuole. The carbon is stripped off by PEP-CK and then fed into the C3 cycle. We look at the timing of rubisco activity. Of course, it's not going to work at night. It's working in the day. PEP-C activity for these PEP-Cs that are involved in CAM photosynthesis are expressed at night. That would be the expectation. The organic acids would show this pattern, increasing at night and decreasing as photosynthesis occurs and uses up that malate mainly during the day and so on. Now, CAM has arisen multiple times in history. Actually, with land plants, there are some lycophytes, for instance, that are doing CAM. Here's agave down here, but there are bromeliads that do CAM. There are orchids that do CAM, and so on. You can see we're actually showing in this figure. We've got both CAM in pink and C4 photosynthesis in blue, and both of those modes of photosynthesis that are both contributing to higher productivity under extreme environments, very hot environments, or very dry environments, or limited environments, have multiple origins across the green tree of life. What John Cushman and collaborators, that they actually had this big DOE Department of Energy project to really do functional work to understand how CAM was working in some monocam species. Actually, agave was one of their species in the agave genome that's been generated. It was generated in association with this project. The other thing that they were able to do using Mesobryanthemum as a model, which is a facultative CAM, the species they work with is facultative CAM. It'll do C3 most of the time, but when it's water stress, it can switch over to CAM. And so they took all of the genes from Mesobryanthemum and put them into a Arabidopsis, and found that they were able to make Arabidopsis plants much larger, even adding many of these things one at a time. And so it's a little bit, they're still kind of scratching their heads. This was in Frontiers in Plant Science, not in Science or Nature, because there's still kind of a puzzle. Exactly how is it doing that? How is introducing the PEP-C or PEP-CK gene from Mesobryanthemum into Arabidopsis, increasing productivity in Arabidopsis? So you can see here's the wild type. Here are all the plants that they introduced these different genes to, MDH, Pepsi K's here, Pepsi's in there somewhere. And they all, each one of them, just adding each of those genes alone and then adding them all together as a group, increase the productivity throughout plants. We've been using the agavoides as a model to understand what genes are really contributing to the evolution of CAM, and how are they, how are they, what's changing in the way those genes are being deployed to facilitate CAM. And this is work that was started in my lab by Carolina Hayduk when she was a student. She's now a PI at University of Connecticut, and she's continuing to do this work. I continue to collaborate with her. So she first noticed that when she made a phylogeny for the agavoides, and then mapped CAM onto that phylogeny, that there seems to be multiple origins of CAM. And in Esperanto, some of the yuccas, not all of them, and in agave and its close relatives. So one thing we would decide to do is take a transcriptomic approach.
So agave, all the agaves are come.
Yeah, there is some variation we haven't looked at, but others have looked at variation, actually developmental variation. So we know seedlings of agave don't do CAM, C3, and in different species are maybe more CAM or less CAM, but all of them have the potential to CAM. There's nothing anywhere you can't get them to do CAM.
Great.
Including Manfredian Polanthes, which are agave now, but they are they are
agave now.
Yeah,
they are also can.
They're not, but they're also yeah. So what we decided to do was go ahead and collect both transcriptome data and physiological data to try to understand, well, what at least in yuccas, what is the difference between the CAM yuccas and the C3 yuccas that might be enabling the CAM yuccas to do CAM and not enabling the C3 yuccas to do CAM. And so here's just very quickly, here's the pipeline for collected samples over a 24-hour period. At the same time, we're collecting the samples, and this is what we're done mostly by Carolina. Rick, another student of mine, helped out later on. I helped out, and then they told me to shoot. It was fast enough. So, they collect those samples, then make RNA-seq libraries that the RNA-seq data, process those data in typical ways. One way, which was not always typical, but makes sense for any sort of screening process, is that we use tools that allow us to identify genes that are showing time-structured expression patterns. At the same time, we grouped all of the genes from these from the C3 species and the CAM species into gene families, and then we could assess how gene expression was changing within a gene family, which paralogs are showing the change in gene expression. And so here we look at our CAM species, our C3 species, and we just look at their gene expression patterns for all the genes that we found in their transcriptome data. And the CAM species has a significantly higher percentage of genes that are showing sometimes signature in their expression. So maybe not so surprising, but when we saw that, I guess I was a little surprised by the magnitude of difference here. There's a lot of genes that are going along with those, that changing their expression patterns and association with CAM. Here are some examples of this sort. So for each of the genes that are showing time signature, we're able to cluster them into particular groups with particular time signatures, that have these various shapes. And now what we're able to do now is go and ask with for the genes within a particular gene family, how do they, their assignment to one of these clusters be different?
Sorry, what is the difference in terms of resistance between these two species?
Resistance to
To
drought. Allifolia is highly resistant. It does slow down in drought, and this, I should say, this experiment was done both under drought conditions and well water conditions. The CAM species is able to deal with drought. It will shut down after a while, but slowly. The C3 species really does it shuts down pretty early, and if it's not gonna, if we didn't start watering again, it would die. Not be watered.
but I mean, in nature,
Yeah.
where these two species are living.
So these are the CAM species is living along the coast in the southeast and in the Caribbean, right on the shore. And so you wouldn't think, well, maybe that doesn't seem like it's a water limited area, but in fact it is. Number one, it's on sandy soils, very well drained, and then it's dealing with sea salt, sea mist all the time, and so it is has less water available to it for transpiration through here. And then the filamentosa lives in the southeast as well, but off the coast under the trees or in open areas that have significantly more water. So they segregate ecologically as you would expect the CAM species in more water limited environment.
Great.
So here we're looking at some of the canonical CAM genes. This carbonic anhydrase, a couple different PEP carboxylases. PEP-CK is modifying the PEP gene, phosphorylating it, which is important for maintaining its activity as the acidity of the cell increases at night in the CAM species. MDH ME, PEP-CK, that's PPCK. These are different things. This decarboxylation and so on. We see that above, we're looking at the CAM species, its pattern of variation. We should kind of pay attention to the both the pattern from day to night and also the axes, the total amount of expression. And we see that in the CAM species here, we're seeing increased expression generally and a shift in time structure. Particularly for, this one actually is the other way around. For this one, it's the other way around. We have increased expression in this one, interestingly. But here we've got, clearly the PEP-C, this particular copy of PEP-C is increasing in expression level. And we've got time structure. Interestingly, the transcripts are made in the evening, in the late day, or even in the early day. And we've got some proteome data here. The proteins are starting to form, are translated later in the day. So transcripts build up, proteins are built, just before dusk, and then those are used up through the night through their activity. This is another PEP-CK, and what's kind of interesting, actually, this one might be doing quite a bit of nighttime carbon fixation. We see that these, the level expression, for instance, for this, but collectively, we look at these three copies, add up to more than the level expression for this copy, the so-called bacterial copy that's typically used for by CAM and C4 species. But here we see that both the C3 species and the CAM species have time structure expression, but it's really ramped up in the CAM species. Same thing here with FCK, and so on. So there's two things going on here.
The sequence of the genes are
Yeah, they're both under
very low.
they're not that different. They're both evolving under constraints, but their expression is changing in one of two ways, either shifting from time structure, from not time structure, just constitutive, to time structured, and or it's just increasing its expression level. So maybe ancestrally they were show this as time structure. It's linked in the circadian clock regulation, but then but it's ramped up in the CAM species relative to the C3 species. So that's something that's I think useful information to understand, you know, if you're interested in biodesign, understanding that, oh, all the building blocks might be there for the CAM species, but you know, the way in which those building blocks are being regulated can vary. And there's a couple of things the way in which we think about them being regulated, or we're making a shift from no linkage to the circadian clock regulatory system to linkage to the regulatory clock system, and then what about the level of expression? You know, just increase expression. Now, something else we've done just to, you know, we for that analysis I just talked about, we needed a yucca genome, and so we've got a very high quality couple of yucca genome sequences that that we have, that we're able to map those reads back to in order to get their gene expression level. The Oak Ridge group has generated a genome for agave tequiliana. And one thing that we did immediately when looking at these genomes is notice that, so here, you know, this is a Synteny plot. We see that there's, you know, these homologous chromosomes in agave and yucca. Maybe here we've got an inversion in one, you know, in one of the two lineages. Here it's pretty much collinearity across the whole thing. There's maybe been a translocation here. But you can see that that there is this high degree of collinearity between these homologous chromosomes. But we look at the off diagonals here, we can see that for each of these yucca chromosomes, we see that there are two other agave chromosomes that show high degree of sensitivity. And we can look in this other direction the same way. Here for this agave chromosome, we see there's not just this, you know, homologous chromosome showing a high degree of synteny. We see that there are you know, two other chromosomes that are showing segments of synteny. So that leads us to infer that the last common ancestor of agavoides, the last common ancestor of agave, or ancestor of agave and yucca, was a hexaploid. So we're working up these analyses.
Jim, just a second. The technology to do that was the assembly of the chromosomes has been done after sequencing, or the chromosomes have been isolated first?
No, we did, extracted high molecular DNA, okay. Made libraries and sequenced using HiFi, PacBio technology, okay. and Hi-C to help bring those into chromosomal assemblies.
Right.
So one thing that's kind of cool about it, one thing that you might have noticed here, or I should point out, is that for both yucca and agave, we've got 5 large chromosomes and a bunch of 25, it turns out, smaller chromosomes. So those are evident up here in this nice micrograph that was generated by folks down here in Brazil. And this has always been a puzzle. How can this group of organisms that have been around for about 27 million years, you know, the last common ancestor of all of the species that show this bimodal karyotype, it was, you know, arose about 25 million years ago. A former student of mine, Michael McCain, you know, did this sort of analysis and make a phylogeny for the group, and then figure out, well, where are the bimodal karyotypes, and where do we have this whole genome triplication? It turns out rather than duplication. And the origin of the bimodal karyotype seems to be correspond to the origin of the polyploid ancestor of agaves and yuccas. Well, it's nice work done by in the University Federal Pernambuco has shown well known that if we do and some analysis that we've done and supported this idea, it doesn't seem like that the big chromosomes have their own special TEs, transposable elements that have accumulated on there. There's transposable elements, all families are representing all chromosomes. It's just these big ones are not getting rid of those transposable elements. So they're really accumulating the big chromosomes, the 5 big chromosomes are accumulating transposons, where the little ones are kind of have the same chromosome set, but they're kind of keeping them in check and not increasing in size. So putting this data together with this data, it's pretty remarkable that you had this genome triplication event occurring, and then very quickly there was this change in the ability of some of these chromosomes to get rid of their transposons as they're always being integrated into the genome. And so these guys very rapidly started accumulating these transposons, getting larger chromosomes, and giving this pattern where really most of the species up in this what we call the bimodal karyotype clade have the same situation: 5 large chromosomes, 25 small chromosomes. There are a couple of lineages here, interestingly, where that's not longer happening, where the bimodal karyotype is starting to go back to being a more single modal karyotype. And so I'm very curious more at these genomes and better understand what happened there. How did these 5 large chromosomes start getting rid of their transposons? One other point I'll make is that we've got the genome assembly that we made is a haploid resolved genome assembly. So we've got full Tesla types, and we found a remarkable amount of structural variation between the 2 haplotypes. And Tequiliana is basically, not quite, basically a clone that was brought into cultivation, and so most tequila, agave tequiliana, is derived from very small, if not one, genotype, and there seems to be this big difference in and it's being propagated vegetatively. There seems to be this big difference in the 2 chromosomes, sets of chromosomes that we find in this tequiliana genome. So, for instance, this is the craziest one, where a big segment of chromosome 2 on this bottom set of chromosomes is moved over to chromosome 3 in this top set, and vice versa. So this is
are both blue agave.
Yeah. This
is
one genotype. We sequenced one genotype, assembled both the maternal and paternal contribution to this genotype as separate assemblies. And so this is in one cell. One cell has got one set of chromosomes that looks like this, one set of chromosomes that looks like this.
Wonderful.
Yeah, it's pretty, and I suspect we're going to find more of this as we generate more haplotype resolved chromosomes. Another thing, if you geek out on this, looking at the distribution of the transposable elements, all these blue segments on transposable elements, you can see really very uniform, pretty uniform across these large chromosomes, less so maybe across the small chromosomes.
They can cross and generate fertile.
Yeah, so I mean, again, this is one genotype. It produces gametes that either have this chromosome or this set of chromosomes, and then cross them if the crosses don't, the growers don't like it because those crosses, it's in particular, it's like hybrid corn. If you make a cross of hybrid corn, you end up with a really diverse population, some of which are okay, many of which are not so good. And this is the exact same thing with Tequila. Yeah. All right, I'm gonna make a shift now to plant sex chromosomes.
Please take your time.
And let's stop. Okay, move on. And because that's another major theme in my lab is understanding the evolution of sex chromosomes in plants. Some of you may be like, what? Sex chromosome plants are the plants I study, like agaves, like, well, all the palms, it's like, the palms that you're working with, are they dioecious or are they producing both male and female flowers, individuals? Do you think they're dioecious? And many palms are dioecious.
Yeah.
And so they have sex chromosomes. And one of the species that has sex chromosomes is Ambarella, which is, it turns out, is sister to all other flowering plants. So we started off sequencing this. We knew it was dioecious. We sequenced it because we were really interested in understanding what was the gene content in the last common ancestor of all flowering plants. So here's Ambarella, here's everything else. By comparing the Ambarella genome to the genome of all of these other things, we can figure out, well, what's the common set of genes? Are there, what about, what is there a common chromosomal structure that we can map back to this last common ancestor of all flowering plants? This is kind of crazy. This is a single species. It only exists in New Caledonia right now. New Caledonia is about 30 million years old. This point is about 150 to 200 million years. So who knows where this last common ancestor was living or where these early members of the Ambarella lineage were. The only place it's found now is in New Caledonia. It lacks wood vessels. So in some ways it's got a wood type that's more like conifers and like other flowering plants. It's got an undifferentiated perianth. So in other words, the petals and the sepals, they don't have that, they just have tepals. That's also thought to be perhaps an ancestral characteristic. But it's dioecious, it can change sex. Individuals can change sex with dioecious, and we were kind of curious to know, well, is that dioecy, is that an ancestral characteristic or is it a derived characteristic? So just looking at the chromosomal structure, one thing is it seems like we determined that whereas many other flowering plant lineages, most of the flowering plant lineages have some plant polyploidy in their history, Ambarella does not. So the last common ancestor of all flowering plants was a tetraploid. We learned from earlier studies, but following its split from Ambarella split from the rest of the flowering plants, it did not have a genome duplication event. Whereas water lilies had one. The eudicots I mentioned, the eudicot was a hexaploid. And in Poaceae, pineapple, for instance, or Poales, pineapple, for instance, it's got evidence for 2 rounds of genome duplication event, one that's shared with our members of Poales, one earlier in monocot history. And so if we take a chromosome from Ambarella and compare it to chromosomes in other species, we can find that there's collinearity between the one Ambarella chromosome in this case and 2 segments of 2 different chromosomes in water lilies. It's a tetraploid, 3 different segments in grapevine because it's ancestrally a hexaploid, and 4 different segments in pineapple because it's had in its history 2 rounds of polyploid. This is work that was done by the Ambarella Genome Project, including Claude Pamphlet, Doug and Pam Soltis, Brad Barberstick, Vic Albert, and Josh Scher and myself. Now, a separate group was working, looking at morphologists, really, were modeling the evolution of floral morphology, and they came to the conclusion that the last common ancestor of all flowering plants was had unisexual flowers, was a perfect flowers, and actually that the evolution of the flowering flower, having perfect flowers, that this was kind of an innovation reproductive part with both male and female function was an innovation with, of flowering plants. And so the dioecious, separate, these are separate genotypes that are produced out of male flowers or female flowers. This is a derived characteristic in Amarella. So we had a mapping population, a population formed by a cross of 2 known individuals, and we, after 2 to 3 years, we had a fair number of those, about 50, 60 plants, between 50 and 60 plants that actually had flowered, and we were able to sex them, and then resequence those individuals and map their reads to our reference genome. And then see, well, where in the genome do we see divergence between the males and the females? And so we didn't know whether this was a XY system like humans or a ZW system where the females that have heterogeneous sex like birds and butterflies. But this data has shown us that on one of these chromosomes, we have this one segment where females were highly heterozygous, males not so much. And that led us to infer that this must be a ZW system, and the sex termination region must be here on chromosome 9. We looked at the genes in this region. We were able to identify Z alleles and W alleles. Z alleles were found in one copy in the female by themselves in the male. W alleles were only found in the females. We were able to identify those for about 90 different genes, make gene trees, and what we found is every one of them showed a pattern like this, where the Z and the W allele were really very similar to each other relative to their homologs sampled from other flowering plant species and even pine, selgium, and moss species. So this again tells us that the Amarella sex chromosome must be young, and again we've got CW system. So this is a derived trait. Also, with these data, we were able to go back and look at, well, how were these reads mapping to the genome, and we found that we didn't have separate Z and W genomes. It wasn't haplotype resolved. And so we determined that helped us see that, oh yeah, this reference genome that we assembled is chimeric in the sex termination region. We've got bits of that assembly where we're only getting reads from the females mapping. And we've got bits of the region where we've got, half of the reads from the females mapping and all of the reads from the males are mapping. So that was, that kind of left us scratching our heads. We really would like to separate the Z and the W chromosomes. And so as part of this Green Genomes project, we went back and did this. And these are the folks at Hudson Alpha that again did the sequencing, the assembly, and kind of led things up. Jeremy Schmutz is the lead of the plant group for JGI, and we got distinct assemblies for haplotype 1 and haplotype 2, fully phased. So we have these female reads that are able to map back to that genome. We had those reads had particular kamers, and we used 41 for this analysis that were only mapping to were only found in the females. And so we were able to map those back then to the 2 haplotypes. So we didn't know in advance which haplotype had a W chromosome, which one had a Z chromosome. But by doing this mapping, it was very clear that haplotype 1 had all these female-specific kamers. Haplotype 2 had none of them. So it's haplotype 1 that has the W chromosome. Something else that this is work done by Sarah Carey, who's a postdoc at Hudson Alpha. Something else that she did was looked at KS in this region, and she finds that there might be 2 strata here. One, the left part of this chromosome has KS that is significantly greater than the right part, and suggesting that that this might be the older part. Okay, so there's been more divergence, maybe more time divergence, for the orthologs that are in this region relative to this region. All right, so that's Amarella. Amarella is a recently derived dioecious species. It turns out that about 6% of flowering plants are dioecious, but they're distributed across 50% of the angiosperm families. So there's been hundreds of origins of dice, and therefore hundreds of origins of sex chromosomes. And this really is kind of a wonderful thing for people who are interested in understanding the evolution of sex chromosomes. The people that are working on mammals, there's one origin of placental mammals, one origin of the separate, X and Y chromosomes, and they're, really can't so the evolution of the process, like the origin, like the evolution of sex chromosomes, when you only have one data point. Now we've got, hundreds of data points representing hundreds of origins of sex termination, including in a number of important crop species. Unfortunately, we only know about which genes are responsible for sex termination in a really handful, 6, maybe 8 now systems. Have we experimentally determined what genes are responsible for differentiating males and females? For Amarella, we don't yet have that. We've got about 90 genes in this region. There are some that have annotations that are very suggestive, but we haven't done any experiments yet to test whether or not those are, in fact, sex termination. Date palm is one where they do know which genes that are responsible for sex termination. So I'd be curious to know in your palm, if they're dioecious, do those same genes seem to be the ones involved in sex termination? Asparagus is one that one of the early ones. Our lab was as part of a PhD project for Alex. We were in collaboration with a bunch of other folks. We were able to identify that there were 2 genes responsible for sex termination, and we started off with our genome sequence. We a double haploid male and a double haploid female. We compared their 2 sequences, identified that their males had a segment of 1 chromosome that had an extra megabase of sequence. We were able to identify the genes that were in that region. And TDF1, just from its, we knew this in the Arabidopsis, we know that it is influences anther development, anther and pollen development. This other gene that turned out to be a sex termination gene, this suppressor of female function, is a Duff gene. So we saw that annotation. We had no idea that it would be involved in sex termination. But it turns out the experimental data suggests that we develop knockouts using gamma radiation and EMS mutations, and then also our Italian friends provide us with spontaneous mutants. And so we have multiple lines of evidence that suggested that in that region, if we knocked out the soft gene, we would convert a male plant to a hermaphrodite plant. If we knocked out in a male plant, knocked out a TDF1 gene, we convert a male to a neuter. If we knocked out the whole region and we had multiple gamma radiation mutants where it just lost all the genes in this region, and doing so would convert that male to a female flower.
You make the radiation, and then after that you look at these.
Yeah. So this was work that was done by these breeders. They had growing plants in Brazil. So one thing about asparagus, you can make YY individuals, and actually the breeders, they make those to develop hybrid XY male lines. A lot of the growers, particularly in Europe, they really like those because they're not self-seeding. If all the seeds male, they don't self-seed in that case. And so we took these all male seeds and irradiated them, and they grew them out in Peru 30 to 40,000 plants in the field, and they were able to just walk through and look which ones are producing fruits. There was a very small number that were producing fruits. Most of them, when we went and sequenced them, had this whole region knocked out. And the flowers, when we looked at the flowers, those were female flowers. There was one that it was just this one gene, this suppressor female function was had a 2 kb missing. And then luckily our backup was one of the our natural gene knockouts, spontaneous mutant from our Italian friends. That same gene had a mutation in a premature stop code. So what we're doing now, so we got that figured out what the sex germination system is in Asparagus officinalis. We knew that there were other dioecious asparagus species in the genus, and we wanted to figure out, and we're still working on figuring out the genes that are sex specific, Y specific in officinalis. Are they Y specific in these other species? As we go down the gene, as we go down this tree. Interestingly, there are these guys that are dioecious, well, or maybe trioecious, maybe they produce hermaphrodites as well. They have a male-specific soft gene, the female suppressor. They don't have a male-specific TDF1. So we're still now trying to figure out, well, what is the male promoting gene that is making it so these individuals can be dioecious. You have males that are suppressing female function and producing pollen. You have females that don't have the suppressor of female function, but they're not producing pollen. So there must be something that's allowing pollen production, driving pollen production in these species, but it's not TDF1 as it is in officinalis. This is work done by a current student of mine, just getting ready to finish up, Phil Betts. Phil took that read mapping that he used to make that inference for soft. He made a gene tree and found that, as we might kind of predicted, that there was a gene duplication event, last common ancestor of those dioecious species, and that one of the copies of those dioecious species is male specific, only found on the Y. So there must have been a gene duplication event followed by neofunctionalization. These gene copies are the ones that are responsible for shutting down female bone. There are paralogs of these or orthologs of these genes in other hermaphroditic species. Obviously, those genes are not shutting down female development. There's another way of looking at it. We've got, and Phil did some molecular dating. We put that soft gene duplication at about 3.5 million years ago, and this clade from this point to this point, they all have a male-specific soft. But it's not until a little bit later, less than 1 million years later, that we see that this TDF1, which is the male promoting gene, becomes male-specific in these dioecious species. And then there's this other dioecious lineage out here that we have no idea. It doesn't have either soft or TDF1 as male-specific. Furthermore, it turns out Taliensis. Here, Taliensis and Cochinchinensis we have as members of this clade. We're working on a genome for both of these species right now. But when we're working on genome, we noticed in the field, and our Chinese collaborators noticed in the field, that there were hermaphrodites, hermaphroditic plants in the population. So there are males, females, and hermaphrodites, all in the same population. It's trioecious. They sequenced the genome of one of the hermaphrodites, and it looked like it's got an XX genotype. It doesn't have soft. It doesn't have that Y-specific soft at all. And it doesn't have really any kind of male or female specific gamers. So it seems to be it seems to have to be all missing the male specific gamers that we're finding in the males of the species. So again, it seems like this species has 3 different sex types. The hermaphrodites are rare, so selection is maintaining dioecy even in the case even with hermaphrodites in the population. So again, we're putting this evolution of dioecy and asparagus into a phylogenetic and geographic context. Most of the all the hermaphroditic species are native to most of them in southern Africa, but they all can get in North Africa as well. And then as range expanded from southern Africa to northern Africa to in this lineage, in this red lineage, to the Caribbean Basin, in this other lineage to Asia, and the first a lot of diversification occurred in what is now Western China. These two lineages that expand out of southern Africa, are the ones that are dioecious. But Hortus seems like it might have expanded out separately from representing this Mediterranean basin species, and it turns out that it's got a completely different Y chromosome. It's also XY. We determine it's the males that have this sex specific gamers, but those sex specific gamers aren't mapping to the Y chromosome homolog in the Hortus, rather mapping to the homolog to chromosome 3. And we're now looking into this region and finding that there's again neither the soft gene nor the CF1 gene. There's about 100 genes in there, and we're now it's just kind of like we have an umbrella. We're looking at the annotations of those genes to try to figure out, well, which ones might be playing a role. Maybe we can use officinalis as an experimental workbench to test the function of these genes. This species is, a wild species is going to be hard to work with experimentally. Officinalis is not so hard to work with experimentally. Stay tuned for that. All this is, we're really kind of curious though, well, are there any common themes? We've got hundreds of origins of dioecy in flowering plants. We've got a few cases now where we know what the sex termination region is. We know what, and can we make any, generalizations? So one generalization that we early on we thought we might be able to make is that older sex chromosomes might be larger than smaller than younger sex chromosomes, but really there's no relationship. See significant relationship between the age of these sex chromosomes and the fraction of the genome that is the sex termination region. One thing that we have seen in the 6 to 8 cases where we've identified the genes is that they're responsible for sex termination. In every one of those cases, there's been a gene duplication and presumably a neofunctionalization. So that may be a common thing, which we're going to have to see as we get more and more genomes, do more and more experimental work on these dioecious species. Just about done. So what I want to pull it all together in the end. A little bit of a stretch, but I think it's not so much a stretch. Alex Harkis is my former student. He now has got a NSF career grant to characterize sex chromosomes across the flowering plant tree of life, and he's made the case that sex chromosomes could be a good target, and maybe this is true for your poem, for putting transgenes to do something interesting. You could, in doing that, you could, know that whatever your process of interest could be sex linked and, improve selection on it. Once it's on, like if it's in the Y region, for instance, it's not going to recombine with the X and escape. So he's made the case for characterizing sex chromosomes and then being able to do something with that by biotechnology, perhaps use optogenetics. So put channel proteins, for instance, or other photochromes into this region, use them as switches for turning things on and off. Folks are already thinking about this, not in the context of sex chromosomes, but generally for controlling somatic behavior and perhaps pushing something to camp.
We are doing exactly this, but with yeasts to construct, to develop a sensor for COVID detection, utilizing exactly the sex system because it's very sensitive. This is a great idea to control stomachs.
So we're in this era now, just like we're entering the genomics era, 10, 15 years ago. Now we're in the kind of biodesign era, where I think we're going to see more. This is really, synthetic plant biology technologies are really emerging. We're going to see a lot of really cool science, really cool biotechnology, and making plants even more useful for the world through this biotechnology. And again, the case that I hope I made to you is that knowing something about biodiversity can help guide us as we're doing this synthetic plant biology going forward. Obrigado.
We have 10 minutes to reach the restaurant. There is no question. Got it. Wonderful. Please, any question? Because I think that I feel that's the beginning of a very important and long-term collaboration. I have a number of ideas. If I stop you as I stopped this, we would never stop the seminar. But a lot of ideas, a lot of things that I see clearly that we are thinking in a similar way in several areas. The things that we are doing to understand transcriptome, to understand diversity by transcriptome. Remember, I would ask you only to send out the seminar.
Yeah.
If you don't.
I guess did it get recorded for your YouTube page, or
It is recorded. It's in YouTube.
Yeah.
If you.
That's fine.
It's public for everybody.
That's fine.
Okay.
Yeah, that's fine. And I'll send the slides as well.
Yeah, please, guys. Good morning.